The first step in understanding brain anatomy is to have a good three-dimensional sense of the structures of the brain, particularly the limbic system. And the first step in this is to have a good sense of the directions of dorsal ventral versus anterior and posterior. So the anterior-posterior axis runs as such. So obviously the spinal cord is the most posterior, and the very tip of the frontal lobe is the most anterior part of the brain. In contrast, the dorsal ventral axis runs like so, dorsal and ventral, and it rotates like this, such that when we come to the frontal lobe, the orbitofrontal cortex here is the most ventral part of the brain, and then we then refer here to the ventrolateral prefrontal cortex and the dorsolateral prefrontal cortex. These terms are used a lot both in neuropsychology and neuroimaging. If I remove one hemisphere, we then can see the medial part of the frontal lobe, and this is the ventromedial prefrontal cortex and the dorsomedial prefrontal cortex, although in many imaging studies this will simply be referred to as the medial prefrontal cortex. Again, you can see its relationship to the orbital frontal cortex, which again is the most ventral structure in the frontal lobe, although the term ventral is often not used. We simply call it the orbitofrontal cortex. Now I'm going to remove this hemisphere, and this gives us a good view of the lateral ventricles as well as the insula. See how the insula sits between the medial temporal lobe and the orbital frontal cortex. Now, as I remove the brainstem from the medial temporal cortex, we can now see the temporal lobe, the temporal lobes, and as part of the medial temporal lobe, we have the hippocampi. The largest or widest part of the hippocampi is the most anterior part, and then it kind of narrows coming back here. When you look at it from this angle, you can see that it resembles a seahorse, which is hippocampus is Latin for seahorse. The white matter comes out and forms the fornix. Fornix is Latin for arch. So here we see the body of the fornix, and then the column of the fornix will be formed here as white matter plunges down to the mammillary bodies. And we'll be looking at that in just a moment when we return to the brainstem. The amygdala would be right here. You can't see it. I'll be showing you that structure in a moment, but just remember that the amygdala is anterior to the hippocampi. Now we're going to take a little closer look at the basal ganglia. Here you see them, it's sitting on top of the brainstem here. I'm going to remove that, and to get a better three-dimensional sense of it, I'll rotate it here for you. Here is the head of the caudate, the tail of the caudate, the putamen. This would be the lateral part, brain, lateral side of the head's over here. Here is the tail of the caudate. This tip at the end is actually the amygdala. I'm going to come back to that in a moment. Now on the medial side of the putamen is the globus pallidus, which you can see here, the pale mound as it is in Latin, just sitting right there, kind of like almost as if it were glued on the medial side of the putamen. Now, when I reinsert this on the brainstem, you can kind of see where it fits. Here are the two thalamic, the two thalami, thalamus here and here. It's shaped somewhat like an egg, with the tip of the egg kind of pointed on the midline. Now you can see how this fits in relationship to the hippocampus. Here is the temporal lobe with the hippocampus. When I reinsert this, you will notice that the thalamus, or the egg of the thalamus, sort of fits right into the curve that the hippocampus makes. Also, you can see that the amygdala here, it's a bit of a tight fit, but as I reinsert this, you can see that the amygdala kind of sits right there, settles right there anterior to to the hippocampus. Now, another final point we'll make in terms of the caudate of the striatum is the relationship of the dorsal and ventral striatum. So obviously here is the dorsal striatum, here is the ventral striatum, and the most ventral part of the striatum, that contains the nucleus accumbens structure that's going to be very important in our study of reward in subsequent lectures. Now we're in a position to discuss Pepes's circuit. So the Pepes circuit begins with information from the entorhinal cortex entering the hippocampus. It's processed in the hippocampus, comes out of the hippocampus through the fornix, down, let me reinsert this here, down the column of the fornix to the mammillary body, and then to the anterior thalamus, and then it goes to the cingulate gyrus, which is that gyrus which is surrounding the corpus callosum. From there, it returns to the hippocampus. This circuit is very important in the modulation of memory and emotion, and we'll be discussing it in great detail in future lectures.